Yes, sir.
Okay. So let's talk about the uh, translation. So I told 3 to 4 different steps. Initiation. First is the activation of tRNA. Second is the attachment of amino acid with the tRNA, that you can say charging of tRNA molecules. And third steps is the initiation. Fourth is the elongation, and fifth is the termination. So now we are going to talk about the termination steps. Okay. So elongation here they have given. Just look at here. During the elongation, I told firstly this ribosomal RNA. Okay, ribosomal RNA, it will move in 5' to 3' direction. Along with this tRNA also will come and it will attach at the acceptor site of ribosome, and those who detach from here, it will go at the exit site. Okay, so in this way, the removal means movement of this ribosome towards the 5' to 3' direction takes place, and elongation steps will complete. Okay, so this is the elongation. Next one, this is also elongation steps, because here tRNA detach and the new amino acid means methionine, they gets attached with the leucine, and new amino acid glycine is coming. Okay, glycine found into the tRNA molecules. This is called the charged tRNA, and this is the uncharged. Uncharged means this type of the tRNA molecules, they don't have the any amino acid. Okay, it don't have any amino acid, so this is called the uncharged tRNA molecules. Clear? So it is called the uncharged tRNA. And upcoming tRNA molecules, this have the phenylalanine amino acid. Means this is the charged tRNA. Okay. So in this way, the 5' to 3' direction, ribosome will move. Along with ribosome, tRNA, they will bring the amino acid and attached at the large subunit of ribosome. And I told one important thing, large subunit of ribosome, it have the ribozyme catalytic activity. Okay, it have the catalytic activity. So this phenomenon is known as the ribozyme. Okay, when the when the ribosome act as enzyme, this is called the ribozyme. The large subunit of ribosome is 23 srRNA. Okay, it acts as a ribozyme. Okay, in this way, the synthesis of polypeptide chain takes place. Again, this is going on, and here one by one, the number of amino acid, their number is going to increase. Here 1, 2, 3, 4, 5, 6. Okay, in this way, and the ribosome also moving into the 5' to 3' direction. This is the elongation, and last is the termination of polypeptide chain. Okay, so termination, I told when it will start, when the nonsense codon. Okay, nonsense or stop codon. Clear? Nonsense or stop codon. Stop codon or terminator codon. Okay, so terminator codon. I told this name, just you can see UAG, UGA, and UA means UAG, UGA, and UAA. Okay, UAA also known as the OCHRE, and UGA it is known as the OPAL. Okay, and UAG is the AMBER. I taught already yesterday. Okay, I taught yesterday these things. Okay, so when it will strike, just read it. The terminator of the polypeptide chain takes place due to the presence of terminator codon, like they have been UA, UGA, and UA. Here, so this is the stop codon. When the terminator codons comes, it does not code for any amino acid. It leads to the termination of polypeptide chain. Okay, suppose that this is the mRNA strand, and ribosome also moving. I told ribosome will move 5' to 3' direction, and it involves in the scanning. When it is moving in this direction, so then suppose that UAA, this stop codon will come. When they will strike with the stop codon, ultimately what happens? Both unit means both subunit of ribosome, it will get detached. Okay, this large subunit and small subunits both got separated. Okay, both got separated. Ultimately, what happened? The formation of polypeptide chain takes place. Okay, formation of polypeptide means polypeptide chain release. Why release? Because there's two subunit of ribosome detached from the mRNA. So in this way, the polypeptide chain which release from the this ribosome. Okay, and it will stop the translation phenomenon. Okay, the polypeptide chain synthesized and released out from ribosome. It undergoes foldage to form specific protein, and the ribosome leaves the mRNA. Okay, when new polypeptide means new protein will form. Okay, this is not mature protein. This is not mature protein. Like mRNA, I told mRNA will form, that is called HNRNA, heterogeneous mRNA. Okay, HNRNA, that is heterogeneous because that mRNA molecules undergo for the post-transcriptional modification. mRNA undergo for the post-transcriptional modification. I told like splicing, capping, tailing, editing. Okay, so similarly, new proteins will form. These proteins undergo also this protein location. So this protein, new protein will form. It undergoes for the PTM, post-translational modification. Post like PTM, post-translational mRNA. So similarly, new proteins will form. This protein is not stable. This cannot perform the function. Okay. So it will be perform the function when the different moiety attached in this protein. Okay. So this phenomenon is known as the post-translational modification. Post-translational modification. Okay. During post-translational modification, HSP, feedstock protein, and glycosidic molecules, glycosidic moiety attached, and some amino moiety also attached. Okay. So ultimately, the protein will form, which is the mature and functional. Okay. When they form, that is called the naive protein. That means it is the new one. They are unable to perform the function. That is naive. Okay. So it will go for the PTM, post-translational modification. If undergo for this process, ultimately they produce the functional protein. Okay, functional protein. This protein can be utilized for the different work in cell. Okay, so this is the story about the termination. Now here you can see this figure, mRNA strands, and this is the stop codon, UGA. When the stop codon strike with the mRNA, strike with the ribosome. The ribosome, large subunit and small subunit detached, and now here detached, and this protein molecule. Now here you can see this protein polypeptide, newly synthesized polypeptide, detached from the ribosome. Okay, and this will go for the post-translational modification. This have again given. Okay, same steps. That ribosome, large subunit, small subunit, and this is the mRNA molecules already decoded. This is the decoded mRNA. This is called the decoded mRNA, and this is the uncharged tRNA. Uncharged tRNA. Okay, ribosome, they have the different subunit. Like ribosome, they have the two. First is the pro, and second is the U. Okay, prokaryote and eukaryote. So prokaryote, they have the two subunit, 50 plus 23s. That will be the 23s rRNA and 16s. Wait one second. Okay, that will be the 23s and 18s. 18s rRNA. Clear? Wait, I'm getting confused. Okay, leave it. Here it will be the 23s you have to learn. I'm forgetting other. So 23s rRNA and the small subunit means 16 srRNA. 16 srRNA. Okay, 16 srRNA also known as the signature sequence. Okay, 16 srRNA, which is known as the signature sequence. Okay, and this is the basis of classification of three domain. Three domain. Fan बंद कर दो, light running. This signature sequence is the basis of three domain classification. Do you know the three domain classification? Anybody, do you know the three domain classification? Anyone, can you tell three domain classification? That three domain also known as the six kingdom classification. Okay, you have to learn six kingdom classification, also known as the three domain. So three domain classification is based on the 16S rRNA sequence. Okay, so they have given the different, you have to learn. I'm telling that prokaryotes they have the 2, large subunit 50, small subunit 30. 50 they have the 2, that is the 23S, 5.8S. Okay, 5.8S, 30S, it will be the 60S rRNA. Clear? Like eukaryotes they have the 2, 60S plus 40S. 60S they have the 25S rRNA, 25S rRNA, 5S rRNA, and 5.8S rRNA. 5.8S rRNA, and 40S means that is the 18S rRNA. 18S rRNA. Clear? So this is the classification of ribosome according to sedimentation coefficient. Clear? According to sedimentation coefficient, they have the prokaryote. Prokaryote is made up of the two subunit, large subunit, small subunit. Large subunit 50S, which have also made up of the two subunit 23S rRNA, 5.8S rRNA, and the prokaryotes they have the 30S subunit, 16S rRNA, and eukaryote 60 and 40. That is the large subunit 60 and small subunit 40. 60 subunit, it is made up of the three component 25S rRNA. I think not 25, I think 28. Sorry, this will be not 25. That is the 28S rRNA. Okay, so 28S rRNA, 5S rRNA. Okay, so 5S rRNA, 5.S rRNA. And last one is the 18S rRNA, which is found into the eukaryote small subunit. Okay, eukaryote small subunit, where the 18S rRNA is only found. Okay, so this is the story about the ribosomal subunit. Okay, so different. So this is the large subunit. Large subunit means 23S rRNA. Clear? And this is the small subunit. If it is the prokaryote means 16S rRNA. Clear? 16S rRNA. So this is the protein synthesis generally takes place in this way. Okay, so this is the overall steps of protein synthesis. Okay, next steps. This is the overall description how they have given. Means ribosome, it is going to synthesize, and this is the 50S subunit means large subunit, and here 30S subunit. That means it is the prokaryote, because prokaryotes they have the 50 and 30. Eukaryotes they have the 60 and 40. So this is the steps by which protein synthesis takes place. Now next topic, and the name of topic is the lag operon. Okay, next topic that you can say lag operon. Clear? Lag operon, or this is also called the gene expression in prokaryotes, or inducible operon concept. Okay, inducible, that is called the inducible operon concept. Inducible operon concept. Okay, or it is known as the lag operon. Lag operon. Clear? Inducible operon concept or lag operon. Clear? Okay. So listen, firstly, just try to clear your concept. What happened? Suppose that this is the test tube. Okay. In this test tube, they have the mixed with the two substance. Here they have the two substance, and here also inside they have the bacteria. E. coli bacteria also present. E. coli bacteria is also present. Here E. coli bacteria is also present. E. coli bacteria is found. Okay. Now this E. coli bacteria, and I told they have the two different types of the medium. How many types? Two different types. So wait. They have the two different types of the media. Just you can see. They have the two different types of the media. First one is the lactose. Red color is known as the lactose, and yellow color is known as the glucose, and white color is the E. coli bacteria. E. coli bacteria. Okay, this is the E. coli bacteria. White color is the E. coli bacteria. When suppose that you are cultivating this E. coli bacteria into this medium, so firstly bacteria, it will try to uptake the lactose molecule. Okay, it will, sorry, not lactose, sorry. It will try to uptake the glucose molecules. Okay, E. coli bacteria firstly uptake the glucose molecules. Okay, uptake glucose molecules. Okay, firstly it will uptake the glucose molecules. Now, okay, this is fine. Ultimately, what happened when glucose is consuming by the E. coli bacteria? The glucose completely consumed, means glucose energy is exhaust, means there is no nutrient media, glucose is absent. Okay, that glucose is absent, means total glucose molecules is completely utilized by the E. coli bacteria. When the glucose exhausts, then bacteria will try to uptake what? It will try to consume the lactose molecules. Okay, it will try to uptake the lactose molecule. Okay, and when glucose is consuming by the E. coli bacteria, then it will suppress the activity of Operon. E. coli bacteria firstly consume the glucose. When it will consume the glucose, it suppress the activity of lactose molecules. Okay, it repress the activity of lactose molecules. So lactose cannot be consumed by the E. coli bacteria. Okay, now when glucose molecules exhaust, what happened? Glucose molecules completely exhaust. When the glucose molecules completely exhaust, so bacteria now that time they don't have any option. So what we will do, it will there is the lactose molecules will come. Okay, what happened? So lactose molecules will come. Okay, and lactose molecules binds with the repressor protein. What is the repressor protein? Repressor protein means this is the protein which inhibit the activity of utilization of lactose. Are you getting or not? Repressor is the protein molecules which inhibit the activity of Operon. Okay, it will binds at the operator site where it can't work. Okay, so this repressor binds there. When lactose molecules will come, suppose that this is the lactose and here this is the repressor. So lactose molecules binds with the repressor. Ultimately what happened? And this is, suppose that this is the active site. This is the active site of lac. Okay, so ultimately what happened? This ribosome, sorry, not ribosome, repressor protein binds with the lactose. It will detach from the operon. Means operator. It will detach from the operator, and ultimately what happen? Operator will be free and they are going to synthesize the other enzyme. Means now they are able to consume the lactose molecules. Okay, now it is able to consume the lactose molecules. So this is the story about the Operon. Operon is the inducible operon concept. Okay, this is the negative regulator. This is the negative regulator. Why negative regulator? Because repressor protein always binds at the operator site. When the lactose molecules will come, it will binds with the repressor detached from the operon, and operator site will be free, and they are going to synthesize the structural gene like XYA, means beta galactosidase and thiogalactosidase, and one more also permease, like permease. Okay, so this is the story. Now you can see Operon concept was first proposed by the French biologist Jacob and Monod. So these two scientists, Jacob and Monod. Just wait. Okay. Who is playing the music? Oh, it is showing. Set up professional audio setting. Okay, leave it. Okay, leave it. So, Operon concept was first proposed by the French biologist Jacob and Monod. Experimentally, they demonstrated that the regulation of gene in E. coli, because I told the gene of bacteria, it is present in the form of cluster. Gene of bacteria, it's present in form of cluster. Cluster means, are you getting? It is present in the form of cluster. Cluster means bunch. That I told already one concept, polycystronic gene. Prokaryotic gene is the polycystronic and eukaryotic gene is the monocystronic. Do you know this thing? I told already in previous classes about this. The prokaryotic gene is the polycystronic, means single gene, it can perform the multiple function, and monocystronic means single gene, single function. So prokaryote, that is the single gene perform more than one function, polycystronic, and monocystronic, single gene can perform the single function. So monocystronic, it is found into the eukaryotes, and polycystronic found into the prokaryotes. So polycystronic gene also you can say it is known as the operon. Okay, it is also called the operon. Is it fine? Okay. Now.
Yes, sir.
Next is these the two scientists, Jacob and Monod. Why it is coming? Just wait, I will remove. Okay. An operon. What is the operon? Just look at it. It is a group of genes that are transcribed at the same time. That is the polycystronic. Okay. The sequential arrangement. Who have some on your mic? Okay. The sequential arrangement of regulatory gene, promoter gene, operator gene, and structural gene in prokaryotes is called the operon concept. Okay. So they have the how many genes? Regulatory, promoter, operator, and S. Okay. So how we'll learn RPOS, regulator, promoter, operator, structural. RPOS. Okay. RPOS. So this is the sequence of gene. Now here you can see this is the sequence of gene. R stands for regulatory, P for promoter, O for operator, and structural gene 3 is there. First one is the LAC-Z, LAC-Y, LAC-A. Okay. LAC-Z, LAC-Y, LAC-A. LAC-Z synthesize the beta galactosidase. LAC-Y synthesizes the LAC permease, and LAC-A synthesize the thiogalactosidase transacetylase. Okay. It synthesizes the three. LAC-Z, beta galactosidase, lactose permease, that is the LAC-Y, and thiogalactosidase transacetylase, it is synthesized by the LAC-A. Okay, so this is called the structural gene. So structural gene have how many components? Structural gene, these have the three components. So first one is the LAC-Z. LAC-Z, they have the enzyme which is known as the beta galactosidase. This is the enzyme which is known as the beta galactosidase. Second is the LAC-Y. It synthesizes the enzyme LAC permease. Okay. And third is the LAC-A. LAC-Z, LAC-Y, LAC-A. LAC-A synthesizes the thiogalactosidase. trans-acetyl. Trans-acetyl. Okay. So these three different enzymes found into means these three genes found into the structural. Okay, these three genes found into the structural gene like LAC-Z, LAC-Y, LAC-A. LAC-Z means beta galactosidase, LAC-Y means lactose permease, and LAC-A means thiogalactosidase trans-acetyl. Okay, thiogalactosidase trans-acetyl. Now look at it. So this is the sequence. This is the sequence you have to learn. What happened normally? Normally they have given Operon. Okay, Operon consists of structural genes each involved in the processing of sugar lactose. One of them is gene enzyme beta galactosidase. The enzyme hydrolyzes lactose into glucose and galactose. We know lactose is the disaccharide. Lactose is the disaccharide. Do you know this thing? Lactose is the disaccharide. This is the disaccharide molecules. When the lactose will hydrolyze, they form the two things. One is the galactose, and one more also glucose. Clear? Lactose will hydrolyze, they form the two components. One is the galactose and another is the glucose molecules. Okay, so this again. One of them is gene enzyme for beta galactosidase, which involves in the hydrolysis of lactose into glucose, and galactose. Clear? Firstly, it will try to utilize the glucose molecules. When the glucose completely exhausts, then it will attack on the disaccharides, because monosaccharides easily digestible, easily can be covered. Okay, so that's why E. coli bacteria firstly it consumes the glucose molecules, then it will go utilize the lactose molecules. Okay, next step, cultural situation of E. coli. I told already here, they have mentioned when the glucose is present and lactose is absent, E. coli does not produce the beta galactoside. I told already, when glucose is present, lactose is absent. Glucose is present, like means no lactose in medium. Okay, okay. So does not produce the beta galactoside. That time enzymes will not, because firstly they have also present. Okay, that means this lactose molecules also present, but it will not utilize the first. Firstly, it will try to utilize the glucose molecules. Okay, so there again, glucose is present, lactose is absent. E. coli does not produce the beta galactosidase. It will not produce the beta galactosidase enzyme. When glucose is absent means exhaust. I told glucose molecules completely exhaust, consumed by the bacteria. The lactose is present in E. coli, produce beta galactosidase. Okay, and I told that this lactose is going to hydrolyze. Okay, this lactose is going to hydrolyze with the help of enzyme beta galactosidase. Beta galactosidase enzyme. It will hydrolyze to the lactose molecules into glucose plus galactose. It will hydrolyze glucose molecules, means lactose molecules, into glucose and galactose. So this is the normal way. Okay, but firstly it will try to consume the glucose molecules. When the glucose completely absent, then it will attack on the lactose. Now this is the operon. This is the operon gene. Here you can see this is the operon. E. coli use either glucose, which is a monosaccharide, or lactose, which is a disaccharide. Lactose needs to be hydrolyzed, digested first, glucose and galactose. I told beta galactosidase enzyme involved. This is the regulatory gene, RPOS, regulatory promoter operator structure. So regulatory promoter operator structure. I told at the operator they have the binds with one protein which is known as the repressor protein. So at this end they have the binds with protein which is known as the repressor protein. What you will say, this is called the repressor protein. Clear? This is called the repressor protein. This is called the repressor protein. Okay, now repressor protein. Okay. Now repressor protein is present here, and when they binds with the operator, the function of structural gene is stopped. structural gene cannot act because operator occupied by the repressor protein. So this is that's why we are saying it is the negative operon. It is the negative operon. Lack operon is the negative and inducible. It is the negative and inducible operon. Okay. When it will not, structural gene not synthesize, it will never consume the lactose molecules. Okay. Look at promoter gene. It is the site for attachment of RNA polymerase second. Okay. That means eukaryotic. Uh, No, we are talking about the prokaryote, so no RNA polymerase. That means simply you can say RNA polymerase second and third, because in prokaryotes they have only one type of RNA, not first and second and third. Okay, here I have written, that is the by mistake, but you have to learn RNA polymerase. Okay, it promotes the structural genes to transcribe through the operator gene. Okay, operator gene to operates and controls the expression of structural gene. Okay, I told operator gene, it controls the expression of structural gene. Means operator is active, structural gene active. Operator is inactive, means structural gene is the inactive. So means operator controls the activity of structural gene. Now same they have given which repressor protein, that is mentioned. Regulator gene, it regulates the operator gene in corporation with the repressor chemical. Okay, this regulator gene, how it will regulate? It synthesize a repressor, and this repressor binds here operator gene. Okay, so structural genes, 3 structural genes are present next to the operator gene. That LAC-Z codes for enzyme beta galactosidase, LAC-Y codes for lactose permeates, and LAC-AZYA, that is codes enzyme thiogalactosidase transacetylase. So these 3 enzymes which involves during the, means they have the structural gene and it involves during the activity of operon. Now, in absence of lactose, I told when in absence of lactose, it will be switch off. Switch off condition. Now look at here. Regulatory gene, no lactose. It will always synthesize a repressor protein, that is LAC repressor. LAC repressor where bind? It binds with the operator region. When the operator region binds, it will stop the activity of LAC-Z, LAC-Y, LAC-A. No transcription, no, that means nil. No transcription means nil. Why? Because this repressor protein binds at the operator region. Okay, so this is the when lactose is not present. Okay, look it here. Same things they have given. Just look it again. Okay, repressor binds, they stop the activity of transcription. So this again. E. coli bacteria that cultured in the absence of lactose media, it would not produce the enzyme beta galactosidase that necessary for the lactose metabolism. Okay, if no lactose in culture media, so they have to stop the activity of lactose utilization. Okay, next step. In this condition, here what happened? When the suppose that what I did, I have used the lactose molecules. This is the lactose molecules. It will get attached with the repressor protein. And when this lactose molecules, that you can say allolactose or lactose. Clear? Allolactose. This is the similar to lactose. Clear? Allolactose or it is similar to the lactose. Either may be lactose or maybe allolactose. Both of them. Okay, either may be lactose or allolactose. When binds with the repressor protein, ultimately what happens? This repressor protein gets detached from the operator region. Okay, when gets detached the operator region, it will be the active. In structural genes, they will start to synthesize. Now look at here. In this condition, structural genes are switched off because the repressor binds with the operator region. In absence of lactose, the repressor protein synthesized regulator gene binds to the operator gene. So now here this repressor protein, it will bind at the operator region, and when the operator region completely binds with this, so it can't perform their activity, means the transcription does not take place. Okay, so transcription does not take place. Next step you can see when the lactose molecules will come. Now here you can see they have not given same things. This blocks the RNA polymerase second to transcribe the structural gene. Hence enzyme are not produced. Same figure. Okay, next step, this have clearly defined. Okay, now look at it. This is the repressor protein binds with the allolactose. When the binds with the allolactose, repressor protein detaches. Repressor protein detaches. Now it will be the active. Means LAC-Z, LAC-Y, LAC-A. So these three enzymes active and they are synthesized to mRNA. That means transcription takes place. Okay, transcription takes place. So this is the overall story about the LAC-Operon concept in prokaryotes. Operon means cluster of genes. In prokaryotic, I told their genes is only found as a operon in the form of bunch, and mRNA strand it is the polycystonic type. Okay, because this is synthesized. Clear? So in this way, when the allolactose binds with the repressor protein, ultimately it will detach from the operator. And now when detached from operator, mRNA synthesized means transcription takes place. Okay, so this is the story about the LAC-Operon. Same concept again they have given. The E. coli bacteria does that culture produce lactose media. Okay, so this is the overall story about the LAC-Operon, and a little bit which have remained I will discuss into the next. So thank you to all of you.